Bioinfocongress.
Dr. Yasin Dağdaş. Selective Autophagy and Cellular Quality Control. Lisans ve yüksek lisans eğitimini Orta Doğu Teknik Üniversitesi Moleküler Biyoloji ve Genetik bölümünde tamamlamıştır. Doktora eğitimini Exeter Üniversitesi'nde bitki ve mikroorganizma etkileşimleri üzerine yapmıştır. Doktor Dağdaş, doktora sonrası çalışmalarını Sainsbury Laboratuvarı'nda gerçekleştirmiştir. 2017'den bu yana Gregor Mendel Enstitüsü'nde grup lideri olarak bitki hücre biyolojisi ve otofajisi üzerine çalışmalarını sürdürmektedir.
Bioinfocongress. Dr. Yasin Dağdaş, Selective Autophagy and Cellular Quality Control. Dr. Yasin Dağdaş completed his undergraduate and graduate studies in the field of molecular biology and genetics at the Middle East Technical University. He completed his PhD at the University of Exeter on the interaction of plants and microorganisms. Dr. Dağdaş carried out her postdoctoral studies at the Sainsbury Laboratory. Since 2017, he has been working on plant cell biology and autophagy as the group leader at the Gregor Mendel Institute.
Bioinfocongress.
Dr. Yasin Dağdaş. Selective Autophagy and Cellular Quality Control. Lisans ve yüksek lisans eğitimini Orta Doğu Teknik Üniversitesi Moleküler Biyoloji ve Genetik bölümünde tamamlamıştır. Doktora eğitimini Exeter Üniversitesi'nde bitki ve mikroorganizma etkileşimleri üzerine yapmıştır. Doktor Dağdaş, doktora sonrası çalışmalarını Sainsbury Laboratuvarı'nda gerçekleştirmiştir. 2017'den bu yana Gregor Mendel Enstitüsü'nde grup lideri olarak bitki hücre biyolojisi ve otofajisi üzerine çalışmalarını sürdürmektedir.
Bioinfocongress. Dr. Yasin Dağdaş, Selective Autophagy and Cellular Quality Control. Dr. Yasin Dağdaş completed his undergraduate and graduate studies in the field of molecular biology and genetics at the Middle East Technical University. He completed his PhD at the University of Exeter on the interaction of plants and microorganisms. Dr. Dağdaş carried out her postdoctoral studies at the Sainsbury Laboratory. Since 2017, he has been working on plant cell biology and autophagy as the group leader at the Gregor Mendel Institute.
Sayın Dr. Yasin Dağdaş hocamız ve değerli katılımcılarımız, hepiniz kongremize hoş geldiniz. Değerli hocamız bugün bizlere Selective Autophagy and Cellular Quality Control konulu bir konuşma gerçekleştirecektir. Konuşma bitiminde ekibimizin hazırladığı ve katılımcılarımızdan gelen soruları hocamıza sunacağız. Tekrardan hoş geldiniz hocam.
Hoş bulduk, çok teşekkürler.
Dilerseniz sununuza başlayabilirsiniz hocam.
Okay. First of all, I would like to really thank to the organizers for inviting me, because it's a real pleasure to be here, and I think it's a really special occasion to be together with, colleagues like yourselves that are excited to hear about the latest scientific discoveries. And what I will try to do today is tell you a little bit about the background and why we want to study autophagy or selective autophagy more specifically, and show you some of the things that we have been doing in the lab. And hopefully in the Q&A session, we can discuss, your interests and kind of our interest more in general in science and also in selective autophagy. But before I start, I would like to kind of hope to invoke some, so that you can think about how the cell is organized, because I think in contrast to what we see in kind of models and drawings, the cell is a lot more complicated. And I'll just start by showing you a video of a 3D volume EM image where they basically very thinly section the cell and then put it together at the end so that we can get a kind of whole picture of the cell. And here we are looking at cells that secrete insulin. And as you can see, it's an extremely crowded environment. So you will see more kind of the separated fractions. So the pink ones here that we see, these little vesicles, are the insulin secreting, insulin containing granules. And then we have the mitochondria, which forms a very dense network because you need a lot of energy, and Golgi because you need to be able to secrete these things, microtubules, centriole, and the big kind of egg in the middle is the nucleus. And as you can see, this does not even include all the things that is happening, for example, ribosomes and all the other cytoplasmic content. And kind of in a way, the cytoplasm looks like this, right? So it is an extremely dense environment. It's a really crowded environment. And what we want to understand, because this is really critical, is I call this the barbecue of the lab. So the big biological question of the lab is to understand how eukaryotic cells very rapidly recycle damaged or unwanted components within the cytoplasm. Because we know that if this process is not happening, in humans, for example, we get all kinds of diseases, including neurodegenerative diseases such as Alzheimer's or Parkinson's. And it is true also for plants. So here you can see two different plants. So this is the wild type plant, which is kind of healthy, and you can see the organs kind of leaves nicely forming. But when you have this quality control mutant, you can see the plants are dwarfed. They are not happy at all. So we want to understand how plant cells within this extremely crowded environment very specifically and selectively basically just pull out the damaged or unwanted components. And for this, we study a process called autophagy. And this comes from Greek. It means self-eating. And this is depicted, this is a kind of cartoon from the Nobel Prize ceremony. It is depicted like this, basically a snake eating itself. And what we have in this case is cellular components being eaten by the cell to be able to maintain cellular homeostasis and adjust to the changing in the environment. And as you all know and probably heard about it, the studies or the discovery of autophagy has been awarded the Nobel Prize in 2016 to Yoshinori Ohsumi, who discovered this process studying yeast as a model organism. And in general, just kind of as an introduction, there are 3 types of autophagy. The first one is, and this will be basically the main focus of the talk, is called macroautophagy, and it has 2 types. One of them is selective, which means it's almost like you are doing surgery. The cell is doing surgery on itself. It's an extremely selective process where you only engulf damaged or unwanted components. For example, here you can see a mitochondria being engulfed with this vesicle, which we will discuss in a minute. Or it can also be non-selective, which is induced upon nutrient starvation conditions. And then you form these double membrane vesicles called autophagosomes. And then these are then delivered to the vacuole or lysosomes for degradation. There are 2 other types of autophagy, just kind of so that you hear about them. One of them is called microautophagy, where the vacuole or the lysosomal or the endomembrane trafficking components basically engulf, without forming these autophagosomes, the cargo. Another one, which is so far has been identified in mammals, is called chaperone-mediated autophagy, where these chaperones, for example, HSP70, that recognizes a specific motif on these aggregate-forming proteins and bring them to the lysosomes. But today, as I mentioned earlier, we will be mostly talking about this process called macroautophagy. I'll just kind of show you a video that very nicely kind of illustrates this process. What you have is an amazing cell biological process where you form a vesicle completely from scratch. So here you have this phagophore, which originates from endoplasmic reticulum. Latest studies have confirmed this. Then it grows by addition of vesicles. So it's basically, you know, it started completely from 0, and then you form this double membrane vesicle called the autophagosome. So this is basically the autophagy organelle that contains these cargo. And then autophagosomes go and fuse with the vacuole or fuse with lysosomes in mammals, and then releases this autophagic body. And then the lytic enzymes in lysosomes or vacuole degrade these contents and then recycle back to the cytosol. So this is basically how autophagy operates. And I'll just show you one real case scenario where we imaged mitochondria autophagy. So it is called mitophagy, autophagy of mitochondria. What you see here is autophagosomes, these autophagy organelles tagged with GFP. So you see them as green. The mitochondria, you can see them as red. So we use a special stain to stain the mitochondria. And then we use chemicals that specifically damage mitochondria. And then we start visualizing these root cells. So these are Arabidopsis root cells under confocal microscope. And what I want you to look at is here in this part. So you can see very, very quickly these, okay, it's not working. Why is it not working? Let's see. Okay, this is not working, but we can look at these electron tomograms, which are basically different parts of this video that's kind of visualized using electron microscopy. So what you have is 3 different steps. So one of them is basically cell that recognizes these damaged components and labels them as non-self. So the cell basically says, okay, this mitochondria is not functioning properly anymore. So I kind of say this mitochondria should be recycled. Then it labels these mitochondria with eat me signals. So these signals are, for example, polyubiquitin chains or changes in the lipid composition, etc. And then you can see autophagosomes basically attacking these eat me signal containing mitochondria and then engulfing them into these double membrane vesicles. There is a 4th step, which I hope I can show you the video. Yes, there is a 4th step, and that is cell type specificity. And what you are looking here is Arabidopsis root cells again, because they are really good for microscopy. And we induce autophagy using a hormone called NAA or auxin, for those of you who are familiar with plant hormones. But what you see here is, even though these 2 cells are adjacent to each other, so these are cells that form root hairs. So these are, you can see the root hairs, and then cells that don't form the root hairs. And you can see these root hair forming cells are full of autophagosomes. And this is basically just a couple of hours. But these cells don't care at all. it is a very cell type specific response, because you can imagine that, for example, a fully differentiated cell or a stem cell will have very different needs in terms of what it needs to recycle, what it needs to degrade and make new compartments, etc. So I think there is a fourth component which is not really well understood in the autophagy field so far, and plants are really good model systems to look into these processes. And I'll just show you two more examples that we are currently studying in the lab. So for example, here is an adapter protein that you can see. So these autophagosomes, these autophagy organelles, are labeled by these ATG8 proteins. You can see they perfectly colocalize. But when we look into a whole root cell, whole root, you can see we see the accumulation in only a few cells. These are the quiescent center cells. So these are basically cells that roots or plants keep kind of in the quiescent state, so that if they need to renew their cells, they can use these cells. Or here is another example which we can visualize using this autophagic flux probe. So autophagic flux means delivery of the autophagic cargo, these organelles or damaged protein cellular components into the vacuole. It's basically a rate probe. You can see, for example, here we are looking at again Arabidopsis roots. In the root cap region, at the tip of the root, we have very high levels of autophagy. But if you cross this, this is wild type cells, and if you cross this to a mutant called ATG5, where we cannot form autophagosomes anymore, as you can see, we completely lose this difference, indicating that again there is very cell type specific or tissue specific autophagy responses, and we need to, instead of kind of grinding whole organisms or basically studying a whole organism, we need to go deeper and look into different cells. And why kind of autophagy become, let's say, famous, and why everyone is interested in studying in autophagy is because it is very much linked to several different types of diseases. We know that autophagy is involved in neurodegenerative diseases, so it is very much linked to aging, Parkinson's, Alzheimer's, etc. Autophagy is also involved in several metabolic diseases, and also it is very much linked to cancer. And that is why there are several kind of startup companies or big pharma companies that are trying to activate autophagy or block autophagy in certain conditions to be able to help curing these diseases. But on the other hand, as you heard at the beginning, we studied plants, and autophagy is very much involved in stress tolerance also in plants. What you are seeing here are three different autophagy mutants. So this vesicle formation requires a whole set of genes. And if you mutate these genes, so these are called autophagy-related genes, ATG genes, you don't form these autophagosomes. And as you can see, when we mutate these genes in Arabidopsis plants and keep them in dark, these autophagy mutants are dying much, much faster than the wild-type plants. So these plants are susceptible to not only darkness, which is basically carbon starvation condition, but all kinds of starvation or abiotic or biotic stress treatments. So autophagy is involved in both health and disease in both, humans or mammalians, and also in plant species. What it really does, and I think this is something that I hope you take from this presentation, is basically it is a cleaning the desk type of response. Just kind of, I have been through that process, and I'm still having kind of nightmares about it. The finals week, right? So when the finals week come, you basically stop everything else, clean your desk, and start working on your finals. And what basically cleans your desk is autophagy. So that is what it does. So just to kind of look at these cartoons, you can see this is a wild type organism where autophagy is functioning normally. You get kind of the warning that it is the finals week, and you clean your desk and turn into this pink final mode. And then as soon as the finals are over, then you get into the party mode. And then again, this becomes, the cell becomes basically in the party mode. But if you look into the ATG mutant, where this cleaning the desk type of responses are not happening, then you end up with chaos. It's basically all green and dark, and that is why autophagy mutants are really, really susceptible. Just to kind of give you a couple of real case scenarios, one of the fascinating features of plants is that they can form a whole organism completely from these stem cells called callus. For those of you who are kind of familiar with plant tissue culture will know these things. So these are these disorganized or unorganized stem cell masses. And then you put them, so this is called callus-inducing media. Then you put them into shoot-inducing media so that plants form these shoots. And then the third stage is to put them into root-inducing media, and then they form their roots. And then you have a whole organism basically differentiated from this. And that is why, you know, plants are amazing organisms in terms of their regenerative capacity. If you look at ATG mutants, because what you do here is basically transition into these shoot or root forming stages, ATG mutants are unable to do this. So they simply cannot kind of reprogram themselves and make these differentiation steps. And this is not unique to Arabidopsis. So here you are looking at a moss called Physkomitrella. And what you see here is when you cut the leaves of this moss, it basically generates these regenerative organs. So it basically kind of forms stem cells at these parts, in the wounded parts. And then you get these cells, or, you know, like the threads of cells called proteinema cells. But if you look at ATG mutants, so here we are looking at two different mutants where they cannot form the autophagosomes, basically cannot clean their desk. And as you can see, they are unable to generate these organs, indicating that we are also looking at an evolutionarily conserved response. So we see a response here in Arabidopsis and also here in Physkomitrella. So one thing that has been bothering us for quite some time is kind of, we are making here a positive correlation based on negative results, right? So we see ATG mutants not being able to do something, and then based on that, we say autophagy is important for this process. Ideally, what we would like to do is identify an autophagy negative regulator, where when we remove that gene, the autophagy process will become faster, and we hope that this will give us some edge on stress recovery. And this is what we found very recently using another moss species called Marcantia. So this is a new model organism in plants. And what you see here is under control conditions, wild type plants, autophagy mutant plant, and this is the negative regulator that we study. What we then do is put these plants into heat stress. So we basically put them in a 37 degree room and then keep them there for a couple of hours and then keep them, move them back to their normal conditions. As you can see in the wild type situation, so kind of normal plants, they start recovering slowly. You can see on the edge that these are kind of, these plants will recover over time. If you look at ATG mutants, they are mostly dead, so they don't recover at all. But when we look at this mutant of this negative regulator, so in this case, we have high levels of autophagy happening in these cells, you can see it is recovering much, much faster. What this means is autophagy is really important for the cellular reprogramming events. And actually, we can use autophagy to be able to hopefully make more tolerant plants to diseases or to different abiotic stress conditions, etc. This is something that we are really excited about, and this is a story that we are following up at the moment. So at the molecular level, we have basically 4 different players. So you have the cargo. So these could be damaged organelles such as chloroplast, endoplasmic reticulum, lipid droplets, mitochondria, or protein aggregates. And then these are recognized by these modular proteins called autophagy receptors. And that is why autophagy process is very, very selective. For example, you would have a receptor that recognizes damaged mitochondria, actually several different types of receptor, or another receptor that recognizes damaged endoplasmic reticulum. That is why the process is very selective. And these autophagy receptors interact with these proteins called ATG8 on the autophagosome membranes. So these are modified by lipids and inserted into this membrane. It's called the isolation membrane. And then that is how they basically bridge the cargo with the autophagosome. So that is how the specificity or the selectivity is achieved. And then you also have other types of proteins called autophagy adapters that are found on the outer membrane of the autophagosome. And then they are important for the trafficking of the autophagosome to the lysosome or vacuole. And as you can see, we have this double membrane vesicle that's called the autophagosome that contains different types of cargo and then it is delivered to the vacuole for fusion. One thing that will be important and that is critical is that autophagy receptors, so these proteins, these two types of proteins, use a short linear motif to interact with these ATG8 proteins on autophagosomes. This motif is called ATG8 interacting motif, or I will say AIM. And what it has, and you can see the crystal structure here, is it is basically docked into the ATG8 via these two loops. One of them is called the W loop because it has this tryptophan residue in the first place. And the other one is called the L loop that you can see here because it has these hydrophobic residues such as leucine, isoleucine, or valine. And that is how the protein, the ATG8 proteins, basically interact with these autophagy receptors. And this motif is conserved from yeast to humans to plant. I mean, it's quite degenerate, but the biochemical feature of this motif is highly conserved throughout all eukaryotic organisms that have autophagy. And this is something that we use a lot in the lab because this gives us a handle to study a protein very specifically in terms of autophagy. If we mutate this motif, if the protein is doing some other functions, we are just basically severing its ties to autophagy and not affecting the whole protein function and whole protein structure. And if you look at selective autophagy receptors, this is an extremely active field of research in general. There are several types of receptors that have been identified and characterized. Just to give you an example, these are all different types of receptors that recognize damaged mitochondria, or these are different receptors that recognize different types of damages on the ER. And when we started the lab, when we looked at what we know so far in plants, this table was almost empty. So that is why we decided to focus our efforts into basically identifying and characterizing these autophagy receptors in plants. And I will, for the rest of the talk, I will basically show you one of these receptors that we have characterized. And as you will see, we can even identify conserved receptor proteins that haven't been identified in humans by looking at them in plants. And this project, the receptor that I will be telling you today is called C53. And the project is driven by two really brilliant PhD students, Madeleine, who just graduated a few weeks ago, and Lorenzo. So this is a protein that has two alpha helical domains on each end. And it is separated, these alpha helical domains are separated by a disordered domain. And as you will know, these intrinsically disordered domains are quite famous these days because they are important for phase separation, or they are really important for organizing protein complexes. What we initially did is we generated CRISPR mutants of the C53 protein, C53 gene in Arabidopsis, because we wanted to make sure that we are not looking at a general autophagy regulator. What you are looking here is two different types of autophagy mutants, and this is an ER stress mutant, IRE1, and I think you heard about it from Elif yesterday. So when we look at carbon or nitrogen starvation conditions where autophagy is really important, you can see these two mutants are dying. So these are core autophagy mutants and they die rapidly. But if you look at C53 mutant, it behaves like wild type. And nitrogen starvation gives us the same phenotype. This indicates that what we are looking here, so the protein that we study is not a general autophagy regulator, but probably has a specific function. But then when we apply ER stress, and in this case, we are using a chemical called tunicamycin, and tunicamycin is a glycosylation inhibitor. So as you may know, glycosylation is happening, so proteins are modified in the ER with this using glycosylation, and this helps them to fold properly. And if you apply tunicamycin, it basically blocks glycosylation and you get ER stress or unfolded proteins accumulating in the ER. When we look at C53 mutant compared to carbon or nitrogen starvation where we didn't see a response, you can see C53 mutants are highly susceptible to tunicamycin. And we are using here IRE1 as a positive control, and ATG mutants are also highly susceptible. One argument which I think is grossly overlooked in the medical field is what we see here could be very specific to Arabidopsis. To make sure that we are not just looking at a one plant response, we also generated CRISPR mutants in Marcantia, so the other model system, the liverwort that I was mentioning before. And as you can see, under control conditions, these plants look fine. So we don't see a clear developmental defect. But when we apply ER stress using this chemical called tunicamycin, you can see C53 mutants are more susceptible. So they are smaller and they are more stressed compared to the wild type. And IRE1 mutant, again, is positive control. So we expect it to be highly susceptible. And again, ATG mutants are basically also susceptible. This indicates that this is a protein that is very specifically involved in ER stress conditions. But then if this is an autophagy receptor, it should be able to interact with these ATG8 molecules on autophagosomes, right? So to test this, we first did cell biology. And what you look here is initially, so we generated plant lines. These are Arabidopsis lines that express stably C53M cherry and also GFP-ATG8. So GFP-ATG8 is basically visualizing or kind of localizing at autophagosomes. Under control conditions, as you can see, these proteins have diffuse localization. So we do not see any puncta or vesicular structures. When we apply starvation, so we basically keep the plants in dark, you can see on the ATG8 channel, so on the green channel, we start seeing puncta. These are basically autophagosomes induced by nutrient starvation. But if you look at C53, we don't see any response. So C53 basically doesn't care if the plant is starved or not. But then if we apply ER stress, we start seeing puncta that colocalize with the ATG8 puncta. So you can see here in the zoomed version, right? So here we have the carbon starvation condition, only ATG8 puncta are forming. But then if you look at the tunicamycin treatment, here we depict it with TM, and you can see these two puncta are colocalizing now. And this indicates that C53 is indeed recruited into these autophagosomes very specifically during ER stress conditions. But then one argument for those of you who are familiar with microscopy, when you see these kind of dots, one criticism, and I think it is a very valid criticism, is that these may be aggregates. So these may be simply because we express these proteins in cells with some text, they may be forming protein aggregates. To make sure that we are not looking at aggregates, what we did is we basically crossed these C53M cherry lines into an ATG mutant. So in the ATG mutant, we do not form any of the autophagosomes. And if they were aggregates, they would still form. But if it is indeed localized to the autophagosomes, then we should not expect to see any puncta. And this is exactly what we see. So you can see under control or carbon starvation conditions, C53 do not form puncta. But when we apply ER stress, we start seeing this puncta formation. But then when we cross it into an ATG mutant where we cannot form any autophagosomes, we do not have any puncta anymore. And this is exactly the case also for ATG8, which is our positive control. So one thing that was kind of exciting for us is that C53 is conserved in all multicellular eukaryotes. But so far, even though people have been studying autophagy in mammalians for 15 years, nobody linked C53 into autophagy with autophagy in plants. So we decided to basically clone the human homologue and then do localization and biochemistry studies to see if also human C53 behaves like an autophagy receptor. So what you are looking here is HeLa cells that express the human ATG8 protein called GABARAP, and then the human C53 homologue with green. And what we look at is basically we look at under control conditions and also this bufilomycin, which blocks lysosomal degradation, and you see additional number of puncta. So because they are delivered to the lysosomes, but then are not degraded. As you can see here, C53 is normally nucleocytosolic. So we see it in the nucleus and in the cytosol, but we don't have any puncta under control conditions. But when we apply ER stress, you can see C53 is depleted from the nucleus and then localizes at these puncta that co-localize with GABARAP. So you can see here in the inset or the zoomed version. And more importantly, these are delivered to the lysosomes, or they fuse with the lysosomes. Because when we apply this bufilomycin, which blocks lysosomal degradation, you can see we get more puncta formation and this indicates that they really behave like canonical autophagy receptors. So what this indicates is that we have identified a new protein using plants that is also conserved in mammalians and behaves like an autophagy receptor. So if you remember, what I mentioned at the beginning is that these autophagy receptors have this ATG8 interacting motifs or aims. So we then decided to test whether C53 has these receptors. For that, we first did a mass spectrometry experiment called native mass spectrometry. What you do here is, instead of digesting the proteins with enzymes like trypsin, you just shoot them straight into the mass spec. You can see, for example, when we shoot C53 into C53 alone, we get these peaks in the mass spec. But then when we mix C53 with human ATG8, we get these additional peaks because they form a complex, and we get these additional peaks due to that complex. And these peaks correspond to C53 interacting one ATG8 molecule or C53 interacting with two different ATG8 molecules. We then did surface plasmon resonance. What you do in surface plasmon resonance, for those of you who are not familiar with the method, is you fix the protein, one of the proteins. In this case, we fix it with we fix ATG8, and then you add your other protein, and then see if they interact, you get these response curves. So you can see as we increase the concentration, we get these additional responses. And more importantly, we can add this AIM peptide. So we just synthesize this peptide and add it to the solution, and then we can reduce the interaction of C53 with ATG8, because this peptide binds to the ATG8 and outcompetes C53. And from this response curve, you can measure the affinity of the interaction. So in this case, for example, Arabidopsis C53 homolog, so AT means Arabidopsis thaliana, interacts with ATG8 with around 1 micromolar affinity. Then, so since we knew that it forms a complex, meaning it directly interacts with our receptor, we decided to find these motifs or these peptides that mediate the interaction. So to do this, what we did is we basically chopped the protein into several different pieces and looked which part of it is important for interaction with ATG8. So here, if you remember, so it has two different domains. We basically generated these different truncations. And what we have seen is when we have this disordered domain, this intrinsically disordered region, we have an interaction. So this is an in vitro pull-down experiment. So we are taking ATG8 with GST, and then we are pulling it down. And then we are looking for this MBP-tagged proteins to see if they interact with ATG8 or not. For example, as a control here, we have GST alone. So if C53 is sticking to the beads, then it would interact with everything, right? So here we can see it is not interacting with GST on its own. But when we have ATG8, we get this interaction. But then when we add this peptide that can outcompete the interaction, you can see we lose this interaction. And then when we look at different truncations, when we have IDR, so N plus IDR, IDR alone, or IDR plus C-terminal, we get this interaction. But if we just basically replace the IDR with a flexible linker, then we have no interaction. This means that this IDR region, this intrinsically disordered domain, is sufficient and necessary for ATG8 interaction. We went one step further and just purified this intrinsically disordered domain from E. coli and performed another biophysical assay called isothermal titration calorimetry. What you do in ITC, so isothermal titration calorimetry, you can shorten it as ITC. You basically mix the two proteins together, and if they interact with each other, they release this heat. You can see the peaks of heat here. And then from these heat peaks, you generate a curve, and the slope of this curve gives you the binding affinity, which again is 1 micromolar, roughly 1 micromolar. And you can also calculate how many of protein A binds to protein B, and similar to the native mass spec experiments, we get 1 C53 binding to 2 ATG8 molecules. And then we spent a lot of time trying to map these motifs because we did not listen evolution. We did not look at the phylogenetic distribution of this protein or did alignments, but instead we did several different mutagenesis experiments. And this kind of cost us several months. So this is another kind of take-home message maybe for those of you who are doing similar experiments. Listen to what evolution has to say, right? So what you are looking here is C53 homologs, alignment of this intrinsically disordered domain in C53 homologs. And we have Arabidopsis, human, C. elegans, zebrafish, Drosophila. So it covers a very huge evolutionary range. But when we align these parts, you can see we have 3 different sites that basically have the same sequence that align with each other. So millions of years of evolution, this protein could have evolved differently. And you can see these parts are completely polymorphic, so we don't get any alignment at all, but these 3 motifs are aligning quite nicely. One thing that was interesting, and this is something that we are continuing to look into, is that this motif is a shuffled motif. So the canonical motif would have the aromatic residue in the first place and the hydrophobic residue in the last place, but as you can see, we have the hydrophobic in the first place and the aromatic in the third place, and that is why we called it the shuffled A. And then we did our mutagenesis experiments. So we basically mutated each of these motifs one by one. So 1, 2, 3. And then in doubles, so 1, 2, 1, 3, and 2, 3. And as you can see, when we mutate all 3 of them, so 1, 2, 3, so the mutation is basically, we are just changing this tryptophan into alanine. So we are not changing the whole motif, but we are just changing these tryptophans into alanines. And you can see when we mutate all 3 of them, we completely lose the binding. And then we complemented, so we basically had a C53 Arabidopsis mutant and complemented this triple mutant, we call it the SA mutant. And as you can see, we don't form any autophagosomes anymore. So we are looking into the number of autophagosomes. In the wild type, we get nice accumulation of autophagosomes. When we complement it again, we get nice accumulation, but in the SA mutant, we don't get any accumulation. And more importantly, it also doesn't complement the ER stress sensitivity. So you can see under tunicamycin conditions compared to the C53 mutant, the wild type plants are growing nicely. And if you put, the wild type complemented one, but if you put this SA mutated variant, we don't get any complementation. And this is not because we are also messing up the 3D structure of the protein, because we measured it using different spectroscopy methods and they fall completely normal. This is really because it cannot take, the C53 cannot localize to the autophagosome. So here you are looking at HeLa cells where we colocalize C53 with GABARAP or the human ATG8. You can see nice puncta forming, but the SA mutant is unable to localize to the autophagosomes. So this indicates that by looking into this protein at the biochemical level, we have identified a new motif that is conserved throughout evolution, and it is really important or crucial for its function. So then the next question, and this is something that kept me awake at night for several nights, is when we apply tunicamycin or different types of ER stress conditions, the stress is happening here in the ER lumen, which is separated from the cytosol with a membrane. So how does this cytosolic protein can sense what is happening in the ER, right? Because what is the connection basically from here to here? We did several different types of biochemical assays and mass spectrometry and cell biology, and we have shown that C53 interacts with an E3 ligase. So it is an E3 ligase that mediates a post-translational modification called eufimylation. I doubt any of you heard about eufimylation, but I am sure all of you heard about ubiquitination. So in ubiquitination, what you have is an E1 enzyme, and then you have this conjugating E2 enzyme, and then you have this E3 ligase that adds this UFM1 or ubiquitin into the substrate. UFM1 is a ubiquitin-like protein, and ATG8 is also a ubiquitin-like protein, by the way. And what we have shown is C53 directly interacts with this UFL1 enzyme on the ER membrane. So that is how it is brought to the endoplasmic reticulum, and that is how it can sense the stress in the ER. Just to show you one piece of evidence about this, so you can see here we have these two proteins, the adapter protein UFL1, forming this colocalizing puncta only during ER stress. But more importantly, if you look at the mutants of these two proteins, C53 cannot form puncta anymore. So what you have is really a receptor complex forming at the endoplasmic reticulum membrane, and it senses and somehow responds to the stress. But then this part of the C53 that mediates the interaction is exposed. So why it doesn't kind of activate autophagy and leads to the eating of the ER all the time? And what we thought is, again, looking at the evolutionary conservation, these two proteins, ATG8 and UFM1, are both ubiquitin-like proteins. So as you can see, when you overlay them, they have similar folding structures. So we thought maybe they are competing with each other. And we did a pull-down experiment. So we are here pulling on C53 and then looking at if it has UFM1 or not. As you can see, under control conditions, basically this is the off state. C53 has UFM1, but when we apply ER stress, you can see C53 loses the interaction. And I'm sure you are guessing what is coming next. And we are looking basically also on the ATG8 case. And as you can see, C53 is not kind of like very weakly interacting with ATG8, but when we apply ER stress, this interaction becomes much stronger. And finally, we kind of looked at where it is integrated into the ER quality control. Yesterday, you heard the talk about these UPR sensors, but what we have seen is C53 is very much linked to these ribosome stalling events. I'll just show you one piece of data. So you can see here two different model substrates. One of them basically leads to the clogging or, stalling of the ribosomes, which means ribosomes colliding with each other. Normally, you know, on one mRNA, we have 10 to 12 different ribosomes simultaneously translating it. But when there's different types of stress conditions, these ribosomes can collide, and this is very detrimental for the cell. As you can see here, we are using a polylysine construct to induce collisions of these ribosomes. C53 in this case is activated very rapidly, and you can see they colocalize with this ERK20 construct. But when we look at another construct that again localizes at these translocons, you can see they form these nice puncta, but C53 doesn't care at all. And this is also very specific to ER-bound ribosomes, because when we generate a ribosome colliding substrate on the cytosol, so it basically only blocks the ribosomes on the cytosols, you can see when we look at vacuolar delivery, we hardly get any puncta in the vacuole. But when we look at this ERK20, where we collide ribosomes on the ER, you can see the vacuole is full of these model substrates. And as you can imagine, we also looked at them in C53 or UFM1 mutant, and we don't see any delivery into the vacuole under in mutants of these guys. So what this really shows is we have used several different organisms. So just kind of to finish off with the model, so we have used Arabidopsis, we have used HeLa cells, we have used another model system called Marcantia. Under normal conditions, when ribosomes are translating happily on the mRNA, this receptor complex is kept inactive by having this UFM1 on the C53. When we have these ribosome collisions, which are very detrimental to the cell, because they do two things. One of them is these incomplete polypeptides, which are toxic to the cell because they start aggregating and they also basically function as sponges to make these toxic environments. Then the ribosomes are modified with this small molecule, small protein called UFM1. And then this leads to the activation of the C53 receptor. And then it basically degrades the toxic polypeptides that will damage the cells. So what we are trying to understand now is how these kind of toxic polypeptides are specifically recruited into these autophagosomes. And we are doing several different experiments looking into cryo-EM structure of this receptor complex, trying to understand how these cargo proteins are selectively recruited, and also trying to understand how UFM1 and ATG8 is working together to regulate this new autophagy pathway. And this is where quite a few people in the lab are focusing on at the moment. With this, I'll finish off and thank to the lab. So here you can see our current group on a Markantia plant. We have several different people working on different autophagy projects, and they are really a diverse bunch. It's a great bunch to work with. And we also collaborate a lot with various people around the world. And I think you heard Elif Karagöz from Max Paris Labs yesterday, and also the funding agencies. And with this, I'll finish here, and I'm happy to take any questions that you may have.
Değerli bilgilerinizi ve çalışmalarınızı bize aktardığınız için çok teşekkür ederiz hocam. Dilerseniz soru cevap kısmına geçebiliriz.
Tabii ki.
Ubiquitin proteazom sistemi ile selektif otofaji arasındaki fark nedir? Hücre kalite kontrolünde ikisinin bir arada çalıştığı ya da ortak etkilediği reseptörler bulunmakta mıdır?
Mükemmel bir soru. Çünkü bizim şu an başka bir projede tam yapmak istediğimiz şey bu. Yakınlarda da bir çalışma yayınladık bununla ilgili. İkisini biz şey diye tanımladık. Brothers in Arms. Yani birlikte çalışıyorlar. Ubiquitin proteazom sistemi ve autophagy aslında birlikte çalışıyorlar. Bir saniye. Şu sunumu durdurursam. Oldu mu şimdi? Sunum tamam. İkisi birlikte çalışıyorlar ve ikisinin birlikte çalıştığı, özellikle protein aggregate oluşturduğunda ikisinin birlikte çalıştığı bir sürü condition var. Bizim düşündüğümüz, şu an için en azından bizim konsept olarak ortaya sunmaya çalıştığımız şey, önce ubiquitin proteazom sistemi gelip kolay sindirilebilecek proteinleri, kolay parçalanabilecek proteinleri parçalıyor. Autophagy sanki böyle şey, eğer gerçekten ubiquitin proteazom sistemi halledemiyorsa, çok büyük aggregate'ler oluşturuyorsa o proteinler, autophagy gelip büyük abi olarak ortalığı temizliyor. Şu an bizim hani datamızın gösterdiği şey bu.
Teşekkür ederim hocam cevabınız için. YouTube'dan bir sorumuz var. Hocam, hücre ölümünün otofajiye bağlı olduğunun kanıtlanması için yapılan çalışmalarda hangi yöntemler kullanılabilir? Teşekkürler.
Hücre ölümü bitkilerde ve hayvanlarda farklı, çünkü hayvanlarda işte caspas'ler ve belli proteinler var ama bitkilerde hücre ölümü karışık bir durum. Çünkü ölen bir hücrede autophagy'nin rolünün olup olmadığını anlamak zor. Nasıl çalışılabilir? Bence çok spesifik hücre ölümüne yol açan proteinler bulunup ondan sonra yine aynı şekilde spatial ve temporal olarak autophagy'yi bloke edebilirsek, mesela o proteinleri aktive ettiğimizde hemen autophagy'yi bloke edersek acaba hücre ölümü oluyor mu olmuyor mu, bu tip deneylere bakmak lazım. Ama sadece autophagy mutantlarında hücre ölümü çalışmak çok zor. Daha kontrollü ve daha daha spesifik deneyler yapmak gerekiyor.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz, sitoplazmanın hacimli kısımlarının stres üzerine bozulduğu durumlarda otofaji seçici olmayabiliyor. Bu süreçte ayrım nasıl yapılıyor?
Bu da çok güzel bir soru. Yani aslında tarihsel olarak autophagy işte seçici, yani işte selective ya da bulk olarak ikiye ayrılıyor. Ama yeni çalışmalar, yeni yapılan çalışmalar aslında bulk autophagy'nin de, yani bu seçici olmayan autophagy'nin de son derece seçici olduğunu gösteriyor. Hücre işte bu mTOR mekanizmasını aktive edip veya işte inhibe edip o şekilde autophagy'yi aktive ediyor ama yine de yani bu seçici olmayan autophagy'de de hücre gidip herhangi bir kısımlarını yemiyor. Yine de belli bir seçicilik söz konusu.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz YouTube'dan bir katılımcımızdan. Öncelikle sunumunuz için kendi adıma teşekkür ederim demiş hocam. ATG8'in aktivasyonu için ATG7 ile kompleks oluşturması gerektiği hastalıklarda terapötik amaçlı hedeflenebilir mi?
Yani bununla ilgili çok fazla çalışma var aslında. Yani işte ATG8 ve ATG7 işte bunların yani bu aslında autophagy'yi aktive etmek ya da inhibe etmekle ilgili şeyler. Bununla uğraşan yani bir sürü startup company'si var. onlar yapılabileceğine inanıyorlar. Ben de hani o kadar güvenmeseler öyle startuplar oluşturmazlar diye düşünüyorum. Muhtemelen yani gerçekten dediğim gibi onlarca company var. Şu anda Autophagy'yi modüle edip terapi amaçlı kullanmaya çalışan, kullanılabilir diye düşünüyorum.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz, otofaji reseptörü modifikasyonlardaki bozukluklar hücresel kalite kontrolde hastalıklara neden olabilir mi?
Evet, dediğim gibi yani şu an mesela Parkinson's hastalığında görevli, yani Parkinson's hastalığıyla çok ilgili olduğu gösterilen birkaç proteinden birinin autophagy ile doğrudan ilgili olduğunu gösteriyorlar. Veya aynı şekilde bu endoplazmik reticulum quality control kısmında görevli proteinlerin farklı işte brain, neural diseases veya metabolic diseases vesaire şeklinde hastalıklarla çok ciddi ilişkisi olduğu biliniyor zaten. Hatta biz çalışmalarımızda şöyle yapıyoruz. O hastalıklarla ilişkili olan proteini hücrede ekspres edip acaba autophagy'de pozitif ya da negatif etkisi var mı ona bakıyoruz aslında. Yani dolayısıyla çok ciddi ilişkililer bunlar farklı hastalıklarda.
Teşekkür ederim hocam cevabınız için. ATG proteinleriyle mikroRNA'lar arasında bir ilişki var mıdır? Otofaji regülasyonundaki rollerinden bahsedebilir misiniz?
MicroRNA'lerle autophagy arasında ilişki var. Yani biz buna çok bakmıyoruz açıkçası ama çalışan gruplar var. Yani bu o proteinlerin seviyesini kontrol etmek için spesifik olarak farklı proteinleri target eden microRNA'lerin olduğu biliniyor. Ama yani bizim çalışmalarımız içerisinde yok bunlar şu anda.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz YouTube'dan. Keratosis pilaris ile otofaji arasında bir ilişki var mıdır? Varsa nasıl bir bağlantı vardır? Gende gerçekleşen bir mutasyon otofajinin etkinliğini değiştirmiş olabilir mi?
Ya bu hastalığı tahminimce melaninle falan ilgili diye, yani deriyle ilgili bir şey olduğunu düşünüyorum. Yani açıkçası hastalığı çok bilmiyorum. Ama eğer düşündüğüm gibi ise hem hani keratin oluşumunda hem melanin oluşumunda veya işte melanin birikiminde autophagy'nin rolü biliniyor. Ama yani bu ayrıntılı şeyini bilmiyorum açıkçası.
Teşekkür ederim hocam cevabınız için. ATG1 ve ATG13 kompleksinin oluşumunu başlattığı fagoforun kaynağı ile fonksiyonu nedir?
Oluştuğu kaynak. Yani şöyle, ATG1 ve ATG13 bunlar geçenlerde yapılan bir çalışmada bunların phase separated compartment'lar oluşturduğunu gösterdiler. Yani bu proteinler sitoplazma içerisinde böyle bir ne denir spesifik bir kompartman oluşturuyorlar ve bu şekilde diğer proteinlerin oraya doğru zamanda doğru şekilde gelip bu vezikülün oluşmasını koordine ediyorlar.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz YouTube'dan. Bitkilerde otofaji tetikler ve etkinliğini arttırırsa Mars ve Ay toprağında bitki yetiştirme zorluklarının üstesinden gelebilir miyiz? Başarılı şekilde yetiştirilenler var ama verimli artırılabilir mi?
Yani ümidimiz o yönde, öyle diyeyim. Yani bu şekilde giderse Dünya'dan en azından Mars'a ya da Ay'a bir şekilde gitmemiz gerekecek ya da başka bir gezegene. Yani az önce o gösterdiğim çalışmalardan birinde bu autophagy'nin negatif regulatorlarını, negatif düzenleyicilerini ortadan kaldırdığımızda bitkiler stresi daha dayanıklı oluyorlar. Ama bu şey değil, ne denir? Yani çok erken safhasındayız bu tip araştırmaların. Moleküler düzeyde bu tip şeyleri anlamadan hani gerçekten bitkileri daha başarılı yapabilir miyiz, daha sağlıklı yapabilir miyiz? Bunları söylemek biraz erken.
Hocam bir diğer sorumuza devam ediyorum. Teşekkür ederim cevabınız için. Klorofajinin parça parça mı, tam bir şekilde mi olacağını belirleyen moleküler mekanizmalar nelerdir?
Bunlar gerçekten çok iyi sorular. Çünkü bizim laboratuvarımızda çalıştığımız konulardan birisi işte kloroplastın nasıl yendiği. Bu hususta şu an itibariyle kimse bir şey bilmiyor. Eğer ilgiliyseniz birlikte çalışabiliriz yani. Ama biz yeni bir reseptör bulduğumuzu düşünüyoruz. Bu reseptörü karakterize ederken işte o sorduğunuz sorunun cevabını alabileceğimizi ümit ediyoruz.
Biz de müthiş hocam. Teşekkür ederiz. Bir diğer sorumuz YouTube'dan. Otofajik organellerde DNA hasar cevabı faktörler arasındaki ilişkileri araştıran çalışmalar sizce neden az sayıda?
Neden az sayıda? Bunun iki sebebi olabilir. Birincisi kimse bakmamış olabilir. Kimse bunu düşünmemiş olabilir. Bu hani olmayacak bir şey değil. Diğeri de autophagy, yani sitoplazmada olan bir şey. Nükleustaki quality control'a ne kadar etkisi var o hususta hani çok fazla bir şey bilmiyoruz henüz. Birkaç tane çalışma var nükleustaki quality control, işte bu içinde görevli olabileceği ya da yardım edebileceğine dair. Ama yani kimse bakmamış da olabilir ya.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz, bitki hücreleri hangi koşullar altında selektif otofajiye ya da ubiquitin proteozoma karar vermektedir?
Bu da çok güzel bir soru. Yani ne denir? Holy Grail sorulardan birisi. Yani dediğim gibi bizim şu an için düşündüğümüz ama bunu yani düzgün bir şekilde araştırmamız gerekiyor. Ubiquitin proteozom ilk işte bu ne denir? Sıkıntılarla ilk uğraşan sistem ubiquitin proteozom. Autophagy sadece ubiquitin proteozom sistemi eğer sorunu çözemiyorsa dahil oluyor. Çünkü autophagy esnasında bir vezikül oluşturmanız gerekiyor ve bu hücre için yani çok pahalı bir olay. Onun için hücre ilk olarak ubiquitin proteozom sistemini kullanıyor. Eğer o yeterli olmuyorsa ya da organellerde bir hasar varsa. Çünkü organeller lipid içerdiği için ubiquitin proteozom sistemi sadece proteinleri parçalayabiliyor. Organellerin parçalanması gerekiyorsa o durumda autophagy dahil oluyor.
Teşekkür ederim hocam cevabınız için. Bitkilerde otofagozom oluşumu memelilerden farklı mıdır?
Temel konsepti aynı. Yani genel itibariyle bu ATG diye bahsettiğim o autophagy related genlerin çoğu aynı. Ama ayrıntılarda farklılıklar var. Ve yani bizim çalışmalarımızın bir kısmı ona yönelik. Acaba tamamen aynı kısımları mı var, farklı kısımları mı var vesaire. Ama temel konsepti, yani sıfırdan bir vezikül oluşturmak ve o vezikülü oluşturmak için gerekli olan işte 20-30 gen, onlar bitkilerde de, hatta yine maya, bitki ve insanlarda korunmuş durumda.
Bir diğer sorumuz hocam. March-Hatiye kullanmanızda sebep nedir? Model organizma olması için ne gibi avantajları vardır?
Bu da çok güzel bir soru. Markantiye'yi niye kullanıyoruz? Çünkü yeast'ten, yani mayadan bilirsiniz, haploid organizma kullanmak her zaman genetik çalışmalar için çok avantajlı. Çünkü bir tane gen var. Onu mutate ettiğinizde fenotip görebiliyorsunuz. O Markantiye'nin size gösterdiğim kısımları tamamen haploid. Ve aynı zamanda genetic complexity dediğimiz, mesela bir genin, işte bir tane protein kinase'in 10 farklı izoformu olabilir organizmalarda. Markantia'da bu diğer bitki model organizmalarına göre bu çok daha basit. Mesela Arabidopsis'te 9 tane ATG8, 9 farklı ATG8 var. Ama Markantia'da sadece 2 tane var. Yani dolayısıyla genetik çalışmalar çok daha hızlı ve çok daha kolay.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz YouTube'dan. C53 diğer retikülofaji reseptörlerine benzemekte midir? Yoksa farklılığı var mıdır?
Yani biz farklı olduğunu düşünüyoruz. Çünkü diğer retikülofaji reseptörleri endoplazmik retikulumun farklı kısımlarını yiyorlar. Ama C53 çok spesifik bir koşulda, yani ribozomların birbirine çarpıştığı koşullarda aktif oluyor. Ve sadece belli endoplazmik retikulumda sentezlenen ve orada fold olan secreted proteinleri parçalıyor. Yani dolayısıyla diğer şeylerden yapısı, en azından fonksiyonu farklı. Ama birlikte çalışıyorlar mıdır diğer reseptörlerle? Bunları daha bilmiyoruz, çalışıyoruz.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz, bitki tümörlerinde otofaji mekanizmasında değişiklik nasıl ve ne yönde olmaktadır?
Bu da yani siz bilmiyorum nasıl bu soruları bu kadar çabuk bulabiliyorsunuz, ama bu da işte benim gelecek 5 yıl için çalışmak istediğim konulardan birisi. Yani geçenlerde bir rapor hazırlıyordum. Aynen bunun üzerine bir proje hazırlıyoruz şu anda yani. Yani cevap olarak bilmiyoruz nasıl olduğunu, ama biz de bunu çok ilginç bulup o hususta çalışmayı istiyoruz.
Hocam önceden hazırlanıyoruz makalelerinizi okuyarak.
Yani ya okumak yeterli değil. Demek ki çok iyi anlıyorsunuz. Gerçekten çok impressive question'lar yani.
Teşekkür ederim hocam platform adına. Bir diğer sorum, otofajik yanıtı düzenleyen ATG proteinleri hangi mekanizmalarla değişmektedir? Bunlar kanserle ilişkilendirilebilir mi?
İlişkilendirilebilir. Yani bu ATG proteinleri çok farklı işte post-translational modification, işte phosphorylation, acetylation, farklı ubiquitination vesaire o tip şeylerle kontrol ediliyor. Ve bu hususlar yani çok ayrıntılı bir şekilde çalışılıyor. Ve kesinlikle kanser veya diğer hastalıklarla çok ilintililer. Çünkü bu mesela phosphorylation olan amino asitler değiştiğinde mesela kansere yol açabiliyor. Yani onun için zaten çok farklı gruplar autophagy ve hastalıklar arasındaki ilişkiyi çalışıyorlar.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz YouTube'dan. Omurgasız hayvanlarda otofajinin metamorfozu yardımcı olduğu görülüyor. Bitkilerde otofajinin metafolojiye bu şekilde ciddi bir oranda etkisi olduğu örnekler var mıdır?
Vardır. Şöyle yani bitkilerde mesela benim gösterdiğim örneklerde o gösterdiğim o fiskomitrelle dediğim yosunda ya da işte markantia'da bu otophagy'nin morfogenesis dediğimiz hani hücrelerin başkalaşmasında çok ciddi rolü olduğu gösteriliyor. Mesela markantia'da çok yeni yapılan bir çalışmada otophagy'nin markantia'nın kamçılı sperm hücreleri var. Otophagy'nin mesela o sperm hücrelerin oluşması için çok yani essential olduğunu gösteren çalışmalar var. Yani o mesela aynı şekilde pirinçte ya da farklı crop organizmalarında otophagy'nin bu değişim için yani bu morfogenesis dediğiniz şeyler için gerekli olduğunu gösteriyorlar.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz, ubiquitin yapısı bozuk proteinleri diğer proteinlerden nasıl ayırabiliyor?
Bu da çok güzel bir soru ve yani senelerdir çalışılan bir soru. Nasıl ayırıyor? Ubiquitinin eklenmesi için de mesela o spesifik E3 ligase dediğim enzimler, bunlardan mesela insanlarda 600 farklı enzim var. Bu 600 farklı enzim bir şey gibi düşünebilirsiniz. Yani bu hücreyi devamlı monitör ediyorlar ve hücrede herhangi bir noktada uygun bir şekilde fold olmamış veya işte bozuk olan bir kısım varsa o enzimler o bozuklukları tanıyıp ubiquitinleri o kısma ekliyorlar ve bu şekilde hani non-self dediğim yani eat me signal olmuş oluyor onlar. Yani beni ye ya da bu proteini ortadan kaldır diye işaretlemiş oluyor.
Teşekkür ederim hocam cevabınız için. Bir diğer sorum, AIM dizisinin ATG8 ile etkileşimini arttırmak için ne yapılabilir?
Bu iletişim için sadece bu core rezidüyü dediğim hani o 4 bir tanesi hidrofobik olan bir tanesi aromatik olan rezidüler haricinde etraftaki amino asit dizisinin de önemi var. Eğer o kısımdaki amino asitler negatif yüklü olurlarsa o interaction daha hızlı oluyor. Yani bizim bu kullandığımız peptit bu C53 ile ATG arasındaki etkileşimi azaltmak için kullandığımız peptit mesela öyle bir peptit. Yani çok sıkı bir şekilde bağlanabiliyor. Çünkü biz o şekilde modify ettik o peptidi.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumuz YouTube'dan. Bitki hastalıklarında selektif otofaji bozukluklarının iyileştirilmesi günümüz gıda sınırlılığında GDO'ya bir alternatif olabilir mi sizce?
Yani şöyle bunları işte o selektif otofaji bozukluklarını iyileştirmek için muhtemelen GDO'yu kullanmak çok daha çabuk olacak. Yani CRISPR vesaire yöntemleriyle bunları modifiye etmek çok daha hızlı olacağı için özellikle mesela Amerika'da veya işte Japonya'da bir sürü firma bu tip şeylerle uğraşıyor şu anda. Hani autophagy veya başka hücresel mekanizmaları CRISPR'la modifiye edip farklı hastalıklara ya da stres koşullarına dirençli bitkiler oluşturmaya çalışıyorlar. Ve çok da başarılı oluyorlar. Bizim hani çalışmalarımızda kullanmaya çalıştığımız bazı şeyler de o.
Teşekkür ederim hocam cevabınız için. Bir diğer sorumla devam ediyorum. Tirozin kinaz inhibitörleri B hücreli kanserlerin tedavisinde otofajik hücre ölümünü hedefleyebilir mi?
Bu çok bildiğim bir şey değil. Yani memelilerde hücre ölümü ve otofeci arasında çok ciddi ilişki var ama autophagy'ye gelmeden çok daha net bir şekilde hücre ölümünü uyaracak şeyler de var. Dolayısıyla hani autophagy'ye ihtiyaç duymadan hücre ölümünü çok daha hızlı induce edecek sistemleri target etmeleri daha kolay olabilir.
Teşekkür ederim hocam cevabınız için. Bir diğer soru. Bitkilerde meydana gelen kuraklık stresi selektif autophagy'ye yönelten bir unsur mudur? Kuraklık stresi altında selektif autophagy ile ilişkili genlerin mRNA profilleri değişiklik gösterir mi?
Kesinlikle gösterir ama ben hani burada bize tartıştığımız için söyleyeyim, ben mRNA değişimlerinden yola çıkma taraftarı çok değilim. Yani bizim burada bir yaşlı bir hocamızın dediği gibi genler iner çıkar. Dolayısıyla bunlardan başlayıp yani çok net bir cevap elde etmek çok mümkün değil. Kuraklık uyguladığımızda autophagy çok hızlı bir şekilde aktive oluyor. Ama bunu işte mRNA profillerine bakarak ne işe yaradığını, ne iş yaptığını anlamamız çok mümkün değil. Daha çok protein ve hücre biyolojisi seviyesinde çalışmaya çalışıyoruz biz.
Teşekkür ederim hocam cevabınız için. Bir diğer soru. Hayvan hücrelerinde meydana gelen otofajik hücre ölümü ile bitki organizmasında mevcut vakuolar hücre ölümü arasında nasıl farklar vardır?
En azından gördüğümüz kadarıyla bitkilerde hücre ölümü çok anladığımız bir mekanizma değil ama gördüğümüz kadarıyla bitkilerde kaspaz dediğimiz o protein grubu olmadığı için tamamen farklı gibi görünüyor. Ama dediğim gibi hayvanlara göre o mekanizma seviyesinde bitkilerdeki hücre ölümünü çok çok az anlıyoruz. Dolayısıyla hani ne kadar farklı, ne kadar benziyor birbirine anlamak çok mümkün değil şu an itibariyle bitkilerdekini bilmediğimiz için.
Teşekkür ederim hocam cevabınız için.
Rica ederim.
Sanırım bu son sorumuzdu hocam. Katılımınız ve verdiğiniz değerli bilgiler için çok teşekkür ederiz hocam tekrardan. Sizi aramızda görmekten teşekkür ederiz hocam. Sizi aramızda görmekten dolayı çok büyük mutluluk duyduk. Gerçekten çok güzel bir sunum ve soru cevap kısmıydı hocam. Size sağlıklı günler dileriz. Görüşmek dileğiyle hocam.
Teşekkürler. Görüşmek dileğiyle. İyi günler.